Mike Riddle holds a degree in mathematics and a graduate degree in education. He served as head of Microsoft's training division for several years. Before getting involved in creation ministry, he was the 1976 U.S. national champion in the pentathlon and served in the U.S. Marine Corps, rising to the rank of captain. Mike is now a full-time speaker for Answers in Genesis. In Cloning, Stem Cells, and Life, Mike addresses the ethical issues surrounding embryonic and adult stem cell research, while at the same time deflating the myths and false reports given by the media. The value of human life is inescapable in his hard-hitting conclusion.
Stem cells, cloning, and life. A lot of misinformation going on around this topic. What we want to do here is try and get through that misinformation and look at what is really happening with stem cells. What about cloning? Is it really working as we're told? And how much do we really value life in this country? So let's start with cloning. What is cloning? Well, first thing we need to understand about cloning, it's really not creating anything new. It's just creating a genetically identical copy. Genetically identical copy. Notice it says genetically identical copy. It means on the outside you might look different. It's only on the inside, genetically, you're identical. It's very much like having an identical twin. So we're really not creating anything new. What we're doing is something called genetic engineering. In other words, all the information is already there. We're just shuffling it around. So we're not creating anything new. How many remember Hello Dolly? It's back in 1996 in Edinburgh, Scotland. They cloned a healthy 6-year-old sheep. That made the press everywhere. Everybody was talking about Hello Dolly. We've cloned a sheep. But what is cloning really all about? See, cloning is all about DNA. We don't create DNA, it is already there. Again, we're doing nothing but genetic engineering, shuffling some things around. Cloning is all about DNA. Depends on your DNA. Now, most of you know that your DNA is made up of all these tight little coiled things called chromosomes. Now, chromosomes, how many of you recognize that you have only 16 unique chromosomes? Anybody just have 16 unique chromosomes? Great, great. We have a few onions here. How many of you have just 6 unique chromosomes? Anybody here have 6? Okay, we have no mosquitoes. How about 24 now? It's the last one on the page. How many have 24? Good, good. We have a few tomatoes here today. How about 78? Now we have a large number. Anybody have 78 unique chromosomes? We have no chickens or hens. How about 40? That's a familiar number. Anybody have 40 here? Okay, we have no mice. How about 46? Anybody have 46 unique chromosomes? Okay, a few of you. Well, you are the only people we have here. If you have 48, you can have a choice. You can either be a banana or you can be a monkey. Oh, but wait a minute, Mike. We do have people with more than 46 chromosomes. That is true. Down syndrome. They don't have 46 chromosomes. They have an extra one. It's a copy of chromosome number 21, but it's still 46 unique chromosomes. It's just an extra copy of one. So they are still human beings. So the number of chromosomes identifies as being human. Now, how did they really make Dolly? Again, it's all about genetic engineering. What we do is take a sheep. We take the DNA or the nucleus out of a sheep egg. So we take the nucleus out of the sheep egg, then we take a cell from a healthy sheep and take the nucleus or the DNA out of that and put it inside the egg. Do a little chemical manipulation and we have a clone. Did we create anything new there? All we did was use existing DNA, shuffle it around to create the clone. Genetic engineering. Now, we heard about Hello, Dolly. How many of us heard about Goodbye, Dolly? See, that didn't make the press a whole lot. At age 3, Dolly was already showing signs of premature aging. At age 6, she died. That's a young age for a sheep. Why did Dolly die of a young age? Well, it's all about your DNA. Something has gone terribly wrong with cloning. It's all about DNA. We all have our DNA. We have our chromosomes. And then on the end of each one of our chromosomes, we have something called telomeres. Now, we look at our DNA. It's commonly thought of about 3% of our DNA is used for coding for making proteins. The other 97% they used to call junk. Oh, it doesn't make proteins, therefore it doesn't have any use. Well, guess what? That was an evolutionist saying. Well, that held up scientific research for many, many years, because we found out that 97% is not junk. We've now identified many parts of it that turn switches on and off. So there is no junk in there. But then on the end of each one of your chromosomes, there's a very special area called telomeres. As far as we know, they don't code for making proteins, and they don't turn any switches on and off. They're like a buffer area between your chromosomes so they don't run into each other. Now, here's what happens. Every time your cells replicate, the DNA gets replicated, but part of those telomeres get lost. In other words, every time your cells replicate, those telomeres are getting shorter and shorter and shorter. And when they get short enough, that becomes a dead cell. In other words, what we have inside of us is something called programmed cell death. In other words, we are programmed to die. I wonder when that might have started. Just a thought. We're programmed to die. We have programmed cell death in our bodies. Illustration here, just an illustration, telomere length and base pairs. Maybe at age 0, when you're first born, your telomeres might be about 8,000 base pairs long. But as those cells, as you grow, the cells replicate, they get shorter and shorter, the telomeres. And maybe at age 35, maybe they're only about 3,000 base pairs. Then as you age more, your cells replicate more. Maybe at age 65, maybe they're only about 1,500 base pairs. So as you continue to age and your cells continue to replicate, the telomeres get shorter and shorter and shorter in those cells. Eventually, they become a dead cell. That's how this process works. Now, more on Dolly. Why did Dolly die so young? Well, first off, it was a 6-year-old cell, the nucleus from a 6-year-old cell that was put into that egg cell. In other words, when Dolly was born, her DNA was already 6 years old. And what did that do to the telomeres? They were already short. That's one of the main problems, big problems with cloning. Now, another thing they don't tell us is how many attempts it took to get Dolly. It took 277 tries to get Dolly. You know what that means by subtraction? 276 failures. They don't talk about those failures very much. So the question I have, if it took that many failures to get Dolly, how many failures is it going to take before we produce a human clone? Now, I know that's been outlawed, but that doesn't mean it has been stopped. How many tries, and what are those attempts going to look like? And if we do ever clone a human being, how long will that human live? And will there be problems? Now, we've cloned a lot of animals. It's not just sheep. We've cloned calves, mice, mules. We've cloned piglets. We've even cloned cats. Every one of the cloned animals has died or is dying a horrible death. That doesn't seem to make the paper. There was a company out there a while back that said you can pay $10,000 to clone your cat. Now, what they don't tell you is that cat on the outside may not even look the same. Here's Cece, the little kitten that was cloned, but look at the mother on the right. Externally, they don't look the same, do they? It's only a genetically identical copy. It says nothing about the temperament of the cloned animal. It might be completely different. But you're going to pay $10,000 to get a cat that might look different from the one you had. Temperament might be different, and high probability it's going to die a horrible death at a young age. Anybody want to pay $10,000 for that? Now, we've done a lot of things with science, and I don't want to discredit science. Genetic engineering is a very good thing. We're doing a lot of things with genetic engineering. We might end up curing a lot of diseases. We might also cause a lot of problems. We just have to be very careful with what we're doing. We might have some future experiments to create some very new kinds of animals. Maybe we'll come up with all this with something called the zebaroo. Who knows? We might be able to create things like that. We might even be able to create the tiger bunnies. How'd you like to have one of those in your house? We might have the birdfish. There's fish with legs right there. No need to wait for evolution. We can probably do that ourselves. And then only a face a mother could love. How'd you like to take one of these home? Now, these are just fictitious, but we are doing a lot of things with genetic engineering. We've actually been able to take the DNA from two different creatures and combine them and come up with a whole new creature. We've been able to do things like that. Genetic engineering is going to solve maybe a lot of problems, but we have to be very careful of what we're doing, because science is good. Why is science good? Because God created all the science. Now let's go back to cloning. There's really two types of cloning we need to talk about. There's reproductive cloning and there's therapeutic cloning. The question is either type of cloning good? Well, let's look what each one does. The goal of reproductive cloning is to produce a baby, as we did in Dolly. Is that good? Well, let's take a look at what the results have been. Every cloned animal has died or is dying a horrible death, usually at very young ages. The goal of therapeutic cloning now is to produce stem cells for research or treatment. So that brings up another topic here, stem cells. What I want to do now is take a detour, talk about these stem cells, then come back to cloning. So stem cells, what are they? They're the cells, they're the body's master cells. Two things they do. They can self-regenerate. In other words, they can make copies of themselves, or they have the ability to differentiate into other types of cells in the body. So those are two things. They're the master cells in the body. But there's two types of stem cells. There are embryonic stem cells and there are adult stem cells. The question there is either one of those good to be using? So let's talk about those. We all begin as an individual cell. Everybody remember that? Okay. Some people have good memory, some people don't. But we all begin as an individual cell. Then as that cell begins to grow, we have a 2-cell embryo. And it continues to grow and we get a 4-cell embryo. Then we continue, we get an 8-cell embryo. How many remember 8 cells? Don't quite remember back to there. Then on day 5, we have something called a blastocyst. We come up with a blastocyst about day 5. Now what's significant about that? Well, it's the blastocyst where we get the embryonic stem cells. Inside that blastocyst we have a thing called the intercell mass, ICM, intercell mass, and that's where we get the embryonic stem cells from. So on day 5 we have embryonic stem cells. Now these embryonic stem cells from the blastocyst, they have the capability of self-renewal. In other words, they can make copies of themselves. They also are what we call pluripotent. They can give rise to the 3 different germ layers, the 3 different layers where we get our adult stem cells from. 3 different layers. In other words, once the first cell is fertilized, it begins to divide and develop into these 3 layers called the germ layers, the ectoderm, mesoderm, and endoderm. This is where we get all the tissues and all the organs from the body from these 3 layers. Now the ectoderm, ecto meaning what? Outer layer. That's where we get things developing such as the brain, the spinal cord, nerve cells, your hair, your skin, and then your sensory organs like the ears and the eyes. Then we get the mesoderm, meso meaning middle or intermediate layer. That's where we get things like the muscles, the blood vessels, and the heart. All develop from that layer. Then we have the endoderm, endo meaning inner layer. That's where we get things such as the stomach, the liver, and the lungs. So the embryonic stem cells develop and we get the 3 different layers, and the 3 different layers is where we get the adult stem cells, which turn into all the different organs and tissues of the body. That's how the process works here. Now one thing about this, we're told the embryonic stem cells, we're told, have the highest capability, highest potential. Why? Because they can differentiate into any kind of cell in the body, become any kind of cell in the body. And that is a good thing, we're told, and it is a good thing, or you may not be here. But embryonic stem cells can become any kind of cell in the body. Therefore, if we can harvest these things, we can maybe rebuild and regenerate tissue that's been damaged from anywhere in the body. Anything that goes wrong, we can fix. Then we're told about these adult stem cells. We're told, well, they don't quite have that potential. They can only become a specific kind of cell in the body, so they don't have all the potential. Well, that's been shown to be wrong. That's been shown to be wrong. See, one of the amazing things about these cells is the adult cells have all the instructions to become anything in the body. It's just that some of them are turned off. Isn't that amazing? How these things just know how to turn things off? But what we have also discovered is from many of these adult stem cells is they can actually become more than one kind of cell in the body. We've now found these adult stem cells have the capability to become almost any kind of cell in the body. Now, do we see any advantage over embryonic stem cells and adult stem cells at this point? Not really. They both seem to have the same capability. Now, here's where we get some difference. The stem cells, or early embryo. What is an embryo? It is a human baby. Got that word? It's an embryo. Or, what's that next word? Disrupted. That's another word for what? Abortion. So the stem cells, or early embryo, are disrupted from natural development through chemical manipulation to become specific tissue types. Here's the word. Expectation is that they will be used to treat unhealthy or diseased tissue. What does that word expectation mean? They haven't done anything yet, have they? It's just that they hope it will be able to do something. I want to show you some deceptive words that are used right now in the media. When they talk about embryonic stem cells, they say, oh, it's not killing a baby, it's not an abortion. So they're using words, it's only a cell, or it's a ball of cells, or maybe it's a pre-embryo. Those are the words they're using to be and they're very deceptive words. They're trying to diffuse this whole issue of what is abortion, what is not abortion. You're not killing anything, it's just a cell, it's a mass. Well, folks, the fact remains that at the moment of fertilization, all the genetic information necessary for its continued development and the mechanisms necessary to transmit that information over the lifetime of the human being are present. Now, as after